So sorry for the interruption in the first video. So here we are continuing our lecture. So here we are discussing about the formation of hepatitis B vaccine, right? So we have discussed about the transformation of hepatitis B surface antigen inside a yeast, right? Now as here we are synthesizing hepatitis B surface antigen in the industrial level, we will simply grow them in fermenter in the absence of leucine. Okay. Now we will isolate the hepatitis B surface antigen particle from whole cell extract. Okay. Now what will be our goal? We will simply transfer them inside the animal to produce the antibody specific for that hepatitis B surface antigen, and we will use those immunoglobulin as a ready-made antibody solution for hepatitis B surface antigen. Okay. So remember, along with yeast we can use Chinese hamster ovary cell, that is a mammalian cell, as a suitable host. Okay. So here hepatitis B vaccine, so it is a sterile solution of immunoglobulin containing antibody to hepatitis B surface antigen that is kept at pH 6.2, and it is formulated in 0.0075 molar NaCl, 0.15 molar glycine, 0.01% of polysorbate 80, and used by intramuscular vein as a vaccine for hepatitis B. Okay. So that is the recombinant vaccine formation. Now we have two topics left, that is from protein engineering. Okay. So we are discuss about that topic after some time. First to discuss about the site directed mutagenesis. I have already made a video regarding site directed mutagenesis. So here we are going to discuss only about site directed mutagenesis briefly, not any very detailed manner. So first of all, it is also known as PCR mutagenesis because here we are using the help of primers. Okay. So point mutation with the help of site directed mutagenesis, that is the main discussion part of us. So I am leaving this because I am going to discuss it with more details. So now how deletion can be incorporated with the help of site directed mutagenesis. So as you can see here we are using two primer, one is forward and a reverse, and those forward and reverse primer are placed in some distance. Okay. As a result, the DNA synthesis will occur in that manner, but that portion will remain unreplicated, right? So as a result, deletion will be incorporated because the nucleotides present, let me clear that picture, present in the normal template will not be further replicated, as here we are not placing any kind of primer, or we are placing the primer in such a way that that portion will become or will remain unreplicated. So that is the idea. And insertion option 2, option 1, here we are inserting a part of nucleotide with the help of site directed mutagenesis. Okay. The primer will simply read it during the DNA polymerase, not primer, sorry for that. The DNA polymerase will simply read that part, okay, during its replication process, and as a result insertion occurs. And this is the insertion option 2. So remember, here we are inserting a stretch of nucleotide at the end of the original primer, right? But here we are inserting stretch of nucleotide in the middle, as you can observe. Okay. So now let's move to the first one, that is how we can incorporate a point mutation, that is also known as, so a site directed mutagenesis, also known as oligonucleotide directed mutagenesis. So here we are modifying protein, okay, at a single amino acid by using a process called site directed mutagenesis. Suppose that we want to change, so as we have discussed it detailedly earlier, we are going to discuss it diagrammatically in a very brief manner. So what is the idea? So we will first construct a plasmid with the help of PBr322 or any other kind of artificial plasmid, where we will incorporate the part or the site where mutation we are willing to incorporate, right? So here we are simply replacing glutamate with aspartate, right? So we will choose a plasmid that will contain the codon for glutamic acid, okay, that is GAA here. So now the idea will be we will separate those double stranded DNA into a single stranded DNA, okay, and we will take the inside the DNA strand present inside under consideration, okay. So let's focus on that part, CTT. Now what we will do, we will simply synthesize a primer, okay, that is specific to the site to be altered. Now the primer will be produced in such a way that it will ensure that after the site directed mutagenesis the aspartate will present instead of glutamate. How? Although the remaining of the primer will be same, in place of C we will incorporate G, in place of T we will incorporate A, but in place of T, that is last one, we will incorporate C, okay. Although it will not form any hydrogen bond between C and T, but as the remaining nucleotide have formed hydrogen bond very well, as a result primer do not found any kind of difficulty to bind with that single stranded DNA molecule, okay. Now this mismatched primer will be used to synthesize a new protein that will have aspartate in place of glutamate. How? Let's see. So along with the modified DNA primer we will also incorporate DNA polymerase, okay. So DNA polymerase will start replication once the primer have successfully annealed with the DNA template, okay. So it will synthesize a new DNA template, as you can see. Now we will again perform the separation step, where now we are taking the DNA strand that is newly synthesized as a template, okay. Then we will perform another step where we are incorporating a short primer, okay, that is just complementary to the 3' end, and then we will incorporate DNA polymerase, so it will again replicate it. As a result we will have newly synthesized double stranded DNA molecule that will, after transcription and translation, leads to the formation of protein that will have aspartate in the place of glutamate, okay. So that is the site directed mutagenesis. So now let's come back here where we are going to discuss about oxidant resistant alpha 1 antitrypsin, okay. So what is the main idea? So remember this, during the respiration we are not only taking air, okay, but we are also taking lot of microbes, okay, that can be infectious, right, because that is why the respiratory tract is the common mode of transmission, right, for many of the diseases, right. For example influenza, right. So influenza virus can insert inside the respiratory tract with the help of the air that we have inhaled. Now the body knows the situation, okay, and that is why it have activated leukocyte and lymphocyte movement there, okay. So during that procedure neutrophil, that is the main culprit here, also enters inside the lungs, okay, and it will create a protein called elastase. This elastase will control the microbial attack, okay, but it have some side effects too. What are the side effects? It will also degrade the elastin present in the lung, that helps in keeping the elasticity of the lung, okay. As a result the elasticity of the lung falls, and which leads to the formation of emphysema, okay. Although our body have a counter defense mechanism for that, That is, in the blood serum, it possesses an enzyme called alpha 1 antitrypsin, a 394 amino acid containing protein which have a methionine in the 358 position and a serine in the 359 position. Okay, as you can see. So now, what is the idea? Idea is, when elastase controlled the microbial attack, alpha 1 antitrypsin will simply inhibit the elastase. Okay, how? The elastase will recognize the 358 methionine and the 359 position serine and bind with that position, and as a result it becomes inactivated because the active side of the elastase is filled with serine and methionine. Okay, and as a result elastase no longer degrade elastin, so the lungs elasticity will not fall. But there is another way by which neutrophil also control microbial attack, that is by releasing reactive oxygen species, a process called oxygen dependent killing of microbes. Okay, that will also destroy microbes, but it has unfortunately another side effect, that is it have the ability to modify the alpha 1 antitrypsin. Okay, how? It will simply replace methionine with methionine sulfoxide, okay, in the 358 position, and which is a bulkier group. As you can see, methionine is obviously less bulkier than the methionine sulfoxide, right? So now this modified alpha 1 antitrypsin now can only inhibit those alpha 1 antitrypsin poorly. Okay, so sorry, I have made a mistake, that those modified alpha 1 antitrypsin can now poorly inhibit the elastase. Okay, so as a result the elastase can now degrade elastin and elasticity of the lung fall, which facilitate emphysema, a situation commonly observed in case of smoker. Okay, so that is the situation that is occurring naturally. So what is the use of recombinant DNA technology here? So as I have discussed about the site directed mutagenesis, so with the help of site directed mutagenesis we will change the methionine with valine, a small codon change. Okay, so now as we have changed methionine with valine, so now that 394 alpha 1 antitrypsin will have valine in the 358th position and serine in the 359th position. So what is the utility? So utility is, this oxidant resistant alpha 1 antitrypsin can inhibit the elastase, okay, as it does earlier, right? But here the utility is, now the reactive oxygen species no longer modifies the alpha 1 antitrypsin, because the reactive oxygen species only have the affinity to methionine. As we have changed the methionine with valine, now it no longer modifies the methionine into methionine sulfoxide, okay, because there is valine in presence of methionine. As a result it cannot make them convert them into a poor inhibitor, and as a result now alpha 1 antitrypsin can easily inhibit the elastase, and as a result lungs elasticity is not altered or not failed. As a result emphysema, the chance of emphysema is reduced. That is the use of recombinant DNA technology in the formation of oxidant resistant alpha 1 antitrypsin. So remember, here with the help of site directed mutagenesis we are simply altering the 358 position methionine with valine. Okay, so that is the idea about the application of recombinant DNA technology. So thank you for listening to this class.